All right, in this next screencast for Chapter 9, we're going to look at the last phase of cellular respiration. Not the last part of Chapter 9, but the last phase of cellular respiration, and this is oxidative phosphorylation. So by this time, we have taken glucose, broken it down to pyruvate over the course of glycolysis, imported it into the mitochondria for the citric acid cycle, the Krebs cycle. And we've pretty much broken glucose down. We've released all of the carbons in the form of carbon dioxide and either made a small amount of ATP via substrate level phosphorylation or transferred the high energy electrons from glucose to electron carriers like NAD plus and FAD. Okay. Now, our final destination for those electrons, okay, is oxidative phosphorylation. Okay, and this is the last step. So there's kind of two processes that are part of oxidative phosphorylation. One is chemiosmosis, and the other is electron transport. And both these two kind of need each other in order to drive the synthesis of ATP. Okay. So as I just said, after glycolysis in the citric acid cycle, all of our high energy electrons are stored in NADH and FADH2. So all the energy from food, which is glucose, is in the form of NADH and FADH2. These two electron carriers will donate their high energy electrons to the electron transport chain. And then that will power the synthesis of ATP via the process of oxidative phosphorylation. Okay, so let's look at a little bit of details here. First of all, where is this happening? So the electron transport chain happens inside or across the inner membrane of the mitochondria. So here we have our outer membrane and then our inner membrane. The electron transport chain is here in the inner membrane. X marks the spot. Okay, so the components of the chain are transport proteins. And these transport proteins are very complex. They're big multi-protein complexes. The carriers, so the proteins in the chain act as carriers, and they alternate between reduced and oxidized forms as they accept and donate electrons down the chain. And this will make sense in a minute when we start to see some of the pictures. As the electrons go down the electron transport chain, they drop in free energy and are finally passed on to oxygen, which, as I said before, is our terminal electron acceptor. And that forms H2O. So this kind of just shows you a little bit about how it works. Up here at the top, and here is my free energy. So at high energy, NADH and FADH2 donate their electrons to these different complexes. There's 4 major complexes in the electron transport chain. Okay. And then these complexes alternate between reduced and oxidized forms. So this is just a series of redox reactions. As they accept electrons, they become reduced, and then they donate the electrons to the next protein. Then that means this one becomes oxidized, and the next one becomes reduced. And they keep passing them on down the chain. As they do, they are releasing energy, and the energy is going to be used by these complexes to pump protons. So these complexes are also proton pumps. And they're going to pump hydrogen ions across the inner membrane of the mitochondria. Okay. And as they do that, the electrons drop in energy until finally they pretty much have given all their energy to the molecules of the electron transport chain. And at the end, they are accepted by oxygen, our terminal electron acceptor, to form water. And again, a lot of the movement of the electrons down the electron transport chain is dictated by oxygen. Remember, oxygen has a very high electronegativity, and it kind of pulls the electrons through the molecules of the electron transport chain towards itself. So this just explains that a little bit more. Electrons are transferred from NADH and FADH2 to the electron transport chain. They are passed through proteins that include these cytochromes, and finally end up in oxygen. Okay. The electron transport chain itself generates no ATP. However, it acts as, again, remember, like a stepwise release of energy from these high energy electrons from the food to the oxygen. Okay. And then all of these multiple smaller steps allow the energy to be released in manageable amounts so we can harness it to do other work. Okay. So the other work that this energy from the electrons does is fuel chemiosmosis. Okay. And this couples the energy from the electron transport chain to ATP synthesis. Okay. And it does this because the proteins in the electron transport chain, as I said, are also proton pumps. Okay. So the electron transfer in the electron transport chain releases energy, and that energy fuels the proton pumps to pump hydrogen ions from the matrix of the mitochondria into the inner membrane space, so across the inner mitochondrial membrane. Okay, this creates a gradient, and we're going to have a picture in just a minute. Okay, now the energy is stored in the gradient. So then, as the protons move back across the membrane, they release energy from the gradient, and they move through, this should be a protein, called ATP synthase. And this, of course, is an enzyme that synthesizes ATP. Okay, so ATP synthase use the exergonic flow of protons, so the diffusion of protons in, taking energy from the gradient, to drive the phosphorylation of ATP. So this is another example of cotransport-ish type thing, where we're coupling the release of energy from the gradient to doing some work. Okay, and this process of energy coupling is called chemiosmosis. And chemiosmosis is the use of energy in a proton gradient to drive cellular work. Okay, and this is a picture of, this is ATP synthase. Okay, and it's the enzyme that makes ATP. So once you have established your gradient, okay, there's a high concentration of protons in the inner membrane space, a low concentration of protons here. The protons flow through ATP synthase, okay, and this changes chemical energy in the gradient actually to mechanical energy. This is a rotor, and the rotor spins, and we'll watch a great video of how this works. Okay, the rotor spins and turns the chemical energy into mechanical energy. This mechanical energy is then used to drive the synthesis of ATP from ADP and phosphate that are in the matrix. So on this is the matrix side, and this is the intermembrane space. Okay, so now kind of let's look at that whole thing. So here, these proteins here are the electron transport chain. You see how they're embedded in the membrane of the mitochondria. And here is ATP synthase. Okay, and as NADH and FADH2 donate their electrons to these molecules, the energy from the electrons is used to pump protons across the membrane. So as the electrons keep getting handed down and down and down, the energy is used to increase the gradient. So to increase the hydrogen ion concentration in the intermembrane space. Okay. Then once the gradient has been established, the protons diffuse back through ATP synthase, releasing energy from the gradient, and that energy from the gradient drives the synthesis of ATP. Okay, so this section here is electron transport, and this section here is chemiosmosis, and together they are oxidative phosphorylation. Okay, so basically that's phosphorylation of ATP using a series of redox reactions. All right, and then this is just kind of a review. The energy stored in the proton gradient couples with the redox reactions of the electron transport chain and causes the synthesis of ATP. Okay, this proton gradient is called a proton motive force because it has the capacity to do work. Okay. The last little bit here I'm going to kind of summarize the ATP production over the course of all of cellular respiration. Okay. During cellular respiration, energy flows in this sequence. It starts off with glucose to NADH, to the electron transport chain, to the proton motive force in the gradient, and then finally to ATP. Okay. So you need to make sure you know this energy flow. Okay. Cellular respiration is really pretty efficient. About 34% of the total energy in each glucose is transferred to ATP, and the end net result ATP production is about 32 ATP. Okay. And the number of ATP is not known exactly, but we can kind of approximate it, and we'll go through the exact calculations in class. And this just kind of shows an overall of the entire process. So we start off here with step 1, glycolysis, okay, where glucose is broken down into pyruvate. Okay, that releases a little bit of energy for some substrate level phosphorylation of ATP, and then puts some energy in NADH. Okay. These pyruvate are transported into the mitochondria for pyruvate oxidation. That process releases some energy, and you get some NADH from that process. The acetyl-CoA is then put into the citric acid cycle, and this is occurring in the matrix of the mitochondria. Citric acid cycle releases some more energy for some substrate level phosphorylation of ATP, as well as additional molecules of NADH and FADH2. So all of these electron carriers carrying all these high energy electrons then take them to the electron transport chain. So the ones from the cytosol obviously have to be moved in. That was fun. Moved in across the membranes. And as you know, this costs a little bit of energy. So it goes across either as 2-NADH or as 2-FADH2. They come in and they get added to the NADH and FADH2 from pyruvate oxidation in the citric acid cycle. They donate all their electrons to the electron transport chain. The energy goes from the electron transport chain into the proton motive force in the gradient. The gradient fuels chemiosmosis, and that fuels oxidative phosphorylation of ATP, and we get a whole bunch more. Okay, so each NADH, one NADH can fuel the synthesis of about 3 ATP, And each FADH2 can fuel the synthesis of about 2 ATP. Okay? So we'll continue to go over this in class, but really make sure that you can trace the energy flow throughout the entire process. Okay, and that's it for cellular respiration.